Welcome back to the No channel. We promised to get back to you on more about the sodium-potassium pump. Let's recap what we know so far. The sodium-potassium pump is a crucial protein found in the cell membrane of neurons. Its primary function is to maintain the proper balance of sodium and potassium ions inside and outside the cell, which is essential for the neuron's ability to transmit electrical signals. The activity of the sodium-potassium pump occurs throughout the entire neuron, but it is particularly crucial in the axon and axon hillock regions. Axon hillock is the region where the axon joins the cell body soma. It is often referred to as the trigger zone because it is where action potentials are initiated. Here's a simplified explanation of how it works. The sodium-potassium pump actively transports ions against their concentration gradients. For every 3 sodium ions it pumps out of the cell, it pumps 2 potassium ions into the cell. The result of this is there are more sodium ions outside the cell than there are potassium ions inside the cell. This difference in ion concentration is crucial for creating the electrochemical gradient necessary for various cellular functions, including the generation of action potentials in neurons. High external and low internal sodium concentration are required for sodium channels to depolarize the neuron rapidly. High internal and low external potassium concentration are required for repolarization. We will get back to this in a moment, but let's just mention a few things first. This active transport process requires energy, which is provided by ATP, adenosine triphosphate. The pump uses the energy released from ATP to change its shape and move the ions across the cell membrane. By maintaining this ion gradient, the pump helps keep the inside of the neuron negatively charged relative to the outside. This difference in charge, known as the membrane potential, is crucial for the generation and transmission of electrical signals called action potentials. Resting membrane potential is approximately -70 millivolts. Most neurons sit between -60 and -75 millivolts when they are not firing. This value is not fixed but depends on the exact type of neuron, the density of K-Pi's leak channels, the local extracellular ion composition, temperature, chloride equilibrium in some cell types. Wait, potassium ion leak channels? These little ones keep leaking out constantly? And this is the interesting part. Potassium ion is about 35% larger than sodium ion, yet in water, the sodium ion binds water more tightly, and together with water, it gets bigger than potassium. And this is super important to understand how mechanically this pump actually works. We will get there. Please note there are also separate sodium leak channels, but there are less of them, so sodium leaks at the slower rate. This means that if the sodium-potassium pump stopped working, eventually all the sodium and all the potassium would leak over time, minutes to hours, and the membrane potential would fall to 0 and the neuron would stop working. Let's try to summarize what we know so far. We have a cellular phospholipid bilayer membrane, which is a great insulator and acts here as an insulator and capacitor. Inside the membrane, we have the leak channels. These passive leak channels are separate for potassium ions and separate for sodium ions. The question is, the channels are passive, and when they open to let go the bigger ion, why the smaller ion does not go through as well? The answer, as is so often the case in life, lies in water. Potassium ions are normally bigger and sodium ions are normally smaller, but this changes in water. In water, the water molecules form a stronger hydration shell around the sodium ion, making this union bigger. Note, this is not chemical bonding to a sodium hydroxide, this is ion-dipole interaction, only physical. The sodium-water attraction is stronger than potassium-water attraction, exactly because sodium ion is smaller, and thus it has higher charge density and stronger electric field, so it attracts the oxygen ends of water, which are negatively charged more tightly. Okay, but in the sodium leak channel, don't we face the same problem, with potassium attempting to escape through it? Yes, we do. For simplified explanation, think this. In the sodium channel, the filtering partially strips the water from the sodium, not completely because that would require too much energy, but just enough to make it go through, while potassium is still unable to pass through. This is a simplification because the filtering does not happen purely by size, and we will explain the selectivity filters soon. Now you may object by saying, hey, how can we call this a passive channel when it uses energy? Valid point, and the answer here is that the channel do not utilize ATP. Instead, the energy for partial dehydration and movement of the sodium ion through the pore comes from binding interactions and the electrochemical gradient. This gradient was achieved by the pump previously, using ATP to bring the ion to the state where it is, so that energy is now only being returned. ATP is the main metabolic energy currency, but it's not the only source of usable free energy in the cell. So even though it uses energy for partial dehydration of the sodium ion, the channel is considered passive because it does not consume ATP. Potassium channel also has its method for dehydrating potassium ion completely, thus making it smaller and easier to pass, while sodium ion remains hydrated. As we mentioned, potassium dehydration is easier because forces holding the water are not as strong as the ion is bigger, and all this is happening further away from its positively charged core. This mechanism for potassium dehydration requires less energy, and it is based on the carbonyl cage. What is carbonyl cage? Chemistry reminder. Carbonyls are organic functional groups characterized by a carbon atom double bonded to an oxygen atom. They are planar neutral but highly polar, where carbon is positive and oxygen negative, and thus reactive. So basically, we have a structure of oxygen atoms, negatively charged, that are attracting positively charged potassium ions, stabilizing them by replacing the water bonds they lost. It is called TVG because of the 5 amino acids it is made of: threonine, valine, glycine, tyrosine, and glycine again. On the other side, in the sodium channel, we have an electrostatic ring, a tight, highly charged funnel called DEK, because of the 4 amino acids it is made of: aspartate, glutamate, lysine, and alanine. Potassium channel is more straight, narrow, and precise, while sodium channel is asymmetric and kind of messy looking. In DEKA channel, the sodium ions flow one by one, never in bulk, while in TVGYG channel, the potassium ions move in a multi-ion stepwise knock-on mechanism. There are 4 binding sites, usually marked S1 to S4, and as a new potassium ion enters the cage, it repels the one ahead, which then repels the next one. Thus, all ions shift one step forward. This process is extremely fast, moving up to 100 million ions per second. Comparably, the continuous flow of sodium ions through DEKA channel one by one is 5 times slower. Please note this: the leak channels are always open, like a dripping pipe. So when we talk about channels opening, we talk about other channels, the voltage-gated channels. More channels! Yes, what is the difference? What is the difference? While they use the same selectivity filters, they have different gating mechanisms. So let's dive into that. Looking at leak channels and voltage-gated channels, what are the similarities and what are the differences? They both use the same selectivity filters, TVG, and DEKA, but leak channels are always partially open, while voltage-gated are closed at rest and open very rapidly when needed. Purpose-wise, leak channels provide a constant baseline permeability, create or maintain the resting potential, and do not participate in the action potential spike, while voltage-gated channels are responsible for the fast upstroke, sodium ions influx, and repolarization, potassium ions efflux. Another similarity is that even when voltage-gated channels fully open during depolarization, ions still pass one by one, never in bulk flow, reaching the speeds we already mentioned. So the selectivity filters, what ions get through, are the same, but the real difference is in the gating when the channel opens and closes, manifested in the presence of the voltage sensors. Besides gating, another difference is that voltage-gated channels are 100 to 1,000 times faster. Voltage-gated channels conduct ions faster, not because of a different selectivity filter, but because of their larger vestibules, stronger electric fields, optimized pore electrostatics, and activation by large voltage gradients, all of which massively increase ion throughput compared to the slow, stability-optimized architecture of leak channels. And despite the greater resistance in the messy sodium channel, less messy than sodium leak channel, though, than in the straight potassium channel, the sodium driving force is greater. Remember what the pump does. There are 3 sodium ions that want to return back to the other side for every 2 potassium ions, plus the potassium ions leak much more, so there is much more built pressure on the sodium side. You can see on the screen the approximate electric currents measured in picoamperes per individual channel. Finally, let's mention there are more voltage-gated channels, millions of them per neuron, than leak channels that number in tens of thousands. This sometimes surprises people because sodium currents feel big and fast. But in terms of actual channel count, potassium outnumbers sodium voltage-gated channels by a large margin, about 10 times more. Same goes for leak channels. There are more potassium leak channels, simply because even a small sodium leak needs to be countered by a larger potassium leak. Also, potassium leak channels come in many subtypes, while sodium leak channels are largely just one type. See the breakdown on the screen, but we will not go into those details. Now, about the gate. Of course, it does not look like on this picture, but how does it look like? Well, like this. The whole channel has a huge voltage sensing machine attached, so-called voltage sensing domain, or VSD. The channel has 6 segments from S1 to S6, and the VSD is formed by S1, S2, S3, and the S4 helix, pocketed among these 3. S4 is the one that moves and opens the gate, so-called pore domain, or PP, at S5 and S6. The whole mechanism is still being debated, so I will keep it as simple as possible, focusing on what everyone agrees on. S4 contains positively charged amino acids. The inside of the membrane is relatively negative, remember, 9 is 70 millivolts, and that charge pulls the S4 inward. When the cell depolarizes, the inside becomes less negative, so this inward pull weakens, and the S4 helix physically shifts outward. This movement is not a slow drift, but when a certain voltage threshold is crossed, could be already at minus 55 millivolts, it snaps rapidly. This S4 movement pulls on S5 and opens the channel gate at S6. Now, there is also this DKA ring, the narrowest part of the pore, acting as the selectivity filter, which picks sodium ions over others, and with its local electric field, helps speed up sodium entry, plus make sure ions move one by one. This whole thing is still more complex than these busy models, and as I mentioned, there are different models that try to explain how exactly this works. I try to show it here to a reasonable level of detail. I hope I managed, at least somewhat. Please note that both voltage-gated sodium and potassium channels use the same basic S1 to S6 architecture. Both use the S4 helix as the main voltage sensor that moves in response to depolarization, but they do not behave identically because of certain differences. Sodium channels have 4 non-identical voltage sensing domains, each tuned differently, so they activate at different voltages, and they are easier to open, requiring only some domains to trigger opening, and then they open very fast, under 1 millisecond. Potassium channels have 4 subunits, each containing 1 VSD, which are the tuned same, activating at the same voltage, and require stronger depolarization and all 4 sensor domains to open, and they open relatively slower, 1 to 5 milliseconds. In conclusion, differences in charge distribution, coupling mechanics, and pore geometry make them activate at different voltages and speeds. This is why sodium channels open first to start the action potential, and potassium channels open later to end it. When it all calms down, the sodium-potassium pump and the leak channels, who constantly keep operating, slowly restore everything back to the initial positions. Now, let's just go back to the action potential cycle to review that entire story, knowing now the details behind it. We have a neuron just sitting there minding its own business, the pump and the leak channels maintaining its resting potential at minus 70 millivolts, keeping fit and ready. When suddenly, a stimulus arrives, but maybe it wasn't strong or lasting enough, so it fails. And again, fail again. But then comes one stimulus that crosses the threshold at minus 55 millivolts, and that's what the neuron was training for, keeping in shape. The mayhem ensues. The S4 helix in the first domain of the sodium voltage gates channel pops, pulls on S5, and the sodium gates open, and sodium ions rush into the cell. Ions going in, we call it influx. This is the stage of depolarization. The repositioning of these positive ions changed the charge differential between the sides of the membrane, and the voltage becomes now positive, and its peak we refer to as action potential. Note that somewhere at -20 millivolts, the S4 helix in the fourth domain of the sodium voltage-gated channel popped and triggered the closing of the channel. This closing is not instant, it is slower than opening, so it will take some time to stop the sodium influx. At about the same time, the S4 helix on all four domains of the potassium channel reached their threshold and they popped too, opening the potassium gates. Again, slowly, so sodium influx still dominated in this phase, until the peak was reached and the potassium ions took over, somewhere around +40 millivolts. So, now the potassium ions in increasingly larger numbers rush out of the cell. Ions going out, we call it eflux. They move in their stepped sequence through the carbonyl cage, slower than the sodium ions rush we just witnessed due to less potential difference and thus less driving force. But they're getting there. We call this repolarization. When does it stop? The concentration gradient is still pushing potassium ions out, but the electrical gradient, now becoming more and more negative again, is pulling the potassium ions in. And when these two forces balance, we have reached the so-called equilibrium potential for potassium, or EK. EK is the membrane potential at which there is no net movement of potassium ions across the membrane, i.e., they are perfectly balanced. And even with the potassium voltage-gated channel still open, the current is zero. This happens at around -90 millivolts. Note, this surpassed the resting potential. That's why we call it hyperpolarization. What happens with the potassium gates that are open? Well, they close when the electric force, now strongly negative, pulls the positive S4 helixes back inwards. They pull on S5 and gates close at S6. Remember, while sodium-gated channels have the S4 helix in fourth domain that helps the sodium gates close quickly, the potassium voltage-gated channels do not have that, and they close slower, which contributes to hyperpolarization, meaning more potassium gets in than needed for the resting state. This is not an inefficiency. There is a plan behind this. The consequence of this is that the neuron is ready to fire again, but not if the same stimulus arrives. Because why would it? If you think of it, that message has already been sent. We got it. No need to repeat. It will fire only if a greater stimulus comes right away that would make it cross the threshold again. That way, the neuron protects itself from unnecessarily firing again and exhausting itself, running out of ammunition or ions. This is called refractory period, a period when a neuron cannot fire another signal. And we divide it in two stages. One, absolute refractory period, which happens due to sodium channels closing and inability of the sodium channel to open again. This lasts usually 1 to 2 milliseconds. And two, relative refractory period, which is caused by the hyperpolarization, requiring greater stimulus, so the firing of the neuron is still possible, but it depends on the stimulus strength. This lasts usually 2 to 5 milliseconds. If no stimulus arrives, the pump and the leaks would restore the voltage to the resting state. Wonderful. We spoke so much about the channels, let's say something more about the pump itself. How does the pump look like? Obviously, it's not a circle and a triangle like in technical drawings and shown here. It looks like this. How does it bind and transfer sodium and potassium ions? The ions are not triangles and squares or whatever is used in model drawings to indicate the concept. So how does the pump pick potassium over sodium ion and vice versa? The pump works through an alternating access mechanism and quite differently than channels. The pump is a transmembrane protein enzyme made of multiple subunits, alpha and beta, that are mandatory, and optional gamma, also known as FXYD. They can further split into different subtypes, but we will not go too crazy in details now. You can see some of them colored here differently. Anyway, the pump splits between the two states using ATP-driven phosphorylation. Phosphorylation is the process of adding a phosphate group from ATP to a molecule, in this case a protein, which changes that molecule's activity or shape. So we have two states, E1 state towards the inside of the cell or cytoplasm facing, with high affinity for sodium ions and low affinity for potassium ions, and E2 state, extracellular facing, with high affinity for potassium ions and low affinity for sodium ions. This flip in protein configuration changes both which side the binding sites face and which ions they favor. And how is that done? Alpha unit performs all transport chemistry. This is the catalytic core responsible for the pump's primary function. It contains the binding sites for sodium ions, potassium ions, and ATP, as well as the phosphorylation site. Beta ensures correct folding and membrane positioning. It inserts the alpha unit in the membrane. Gamma modulates pump kinetics. It changes the pump's affinity for sodium, potassium, and ATP. Wait a second. If the change in affinity is the key for the pump to function, and gamma subunits affects the affinity, how come the gamma subunit is optional? Well, let's be very accurate here. It affects the affinity, but that does not mean there is no affinity without it. Without gamma subunit, the pump still works. Alpha and beta are sufficient, but it may operate suboptimal. If you think of alpha and beta as an engine inside a car, then gamma is the engine tuning chip that boosts its performance. Gamma is present where it is super important to regulate pump efficiency, like in a heart that needs to pump faster or slower depending on adrenaline, or kidneys that need to adjust sodium affinity based on varying salt load. Evolutionary, alpha is the oldest, providing core machinery across all life. Then, as the cells in their membranes evolved, the beta emerged, about 2 billion years ago. Beta is found only in eukaryotes. Finally, gamma, the engine tuning chip, is the most recent innovation, around 500 million years ago, unique to vertebrates who require high cardiac performance, complex renal salt balance, advanced neuronal firing precision, and tight metabolic regulation. Back to changing states E1 and E2. Important detail here is that ATP is only consumed during the E1 to E2 transition, phosphorylation. E2 to E1 transition does not require ATP. It is driven by dephosphorylation and ion binding. In the E1 or inside-facing state, this happens. The pump binds 3 sodium ions, ATP is hydrolyzed, and the pump becomes phosphorylated. This phosphorylation provides the energy needed to change conformation, open outward E2 state, and release sodium ions to the outside. ATP is consumed only here. This is the active step. Question: how does the pump bind 3 sodium ions? Meaning, what is the exact mechanics of it? And how come it does not accidentally grab a potassium ion? Remember, there are no triangles and squares and whatnot. The cavity contains 3 partially overlapping cation sites, classically termed sites 1, 2, shared between sodium and potassium ions, and a sodium-specific site 3. Binding is sequential and cooperative. Sodium at one site slightly rearranges helices and side chains to form the next site with higher affinity. Here's how it happens. The sodium ion first occupies the site 3, which sort of pushes the helix M4E nearby, which is now configures site 1 to accept smaller sodium ions rather than potassium ion, which then pushes helices around site 2 to configure it for sodium ions only state. So there is a size constraint. The sites are located in a pocket between alpha TM34 subunit and alpha TM510 subunit, specifically between helices 4, 5, and 6. Additionally, there are certain acidic residues that get positioned so they favor sodium, or in a different position, potassium. So when positioned to favor sodium, if a potassium ion shows up, the binding is weak and unstable. And finally, there is a quality control mechanism. If a wrong ion enters the site, the pump cannot proceed with the next step. How does that happen? We introduce occlusion. Occlusion is a tight mechanical cap over the site with the right ion in. If covering the ion in the site requires too much energy, and so is the case if the wrong ion is in, the occlusion does not happen, meaning that the ion is not trapped by the membrane and remains available to cytosol. And since the binding of that ion is weak, it will eventually leave the site back to cytosol, opening the space for another ion to attempt nesting in the site. Once the right ion is in, the occlusion can occur with the acceptable level of energy, and the next step can happen. Quick side note, what are exactly these acidic residues we mentioned? Amino-acids. Among the 20 standard amino acids, 2 are acidic: aspartic acid, ASP, and glutamic acid, GLU. These residues are acidic because in water, which is neutral, pH equals 7, their side chains tend to lose a proton, leaving a negatively charged carboxylate, CO-, and that's why they pull the positively charged sodium and potassium ions. Sounds silly though. Out of 20 amino acids, only 2 are acidic. Why we call them acids then if they are not acidic? To learn more about that, stay tuned for our video about amino acids. It's still in the making. Side note end. Now let's see the E2 state. In the E2 or outside-facing state, this happens. The pump binds 2 potassium ions, and the phosphate group is released, dephosphorylation. Dephosphorylation triggers the return to E1. No ATP is consumed. This step is energetically downhill. E2 to E1 is therefore the passive step. It's like a mechanical spring restoring its state after tension or pressure. Okay, so now going back from E2 to E1, how come that the smaller sodium ion does not occupy the space of the larger potassium ion? Mostly due to chemistry. The before-mentioned acidic constellations change and start favoring potassium over sodium, so sodium ions now bind weakly and leave the sites, and potassium ions come in and bind tight. Remember, potassium ions cannot enter the site 3 because it is too small for them. What about the occlusion? Well, that changed too. With the new configuration, it requires less energy to close over potassium ion than sodium ion. When all are ready, the pump can spring back to E1 state and start shifting again, fueled by ATP. I think we should end it here. This is all so fascinating, and as you could see, there is so much more to talk and to discover, and I invite you to keep exploring. Thank you for now and your desire to know. If you enjoyed this, don't forget to like, share, and subscribe to stay tuned for more updates.